Imagine asking an AI to find a possible drug candidate. And instead of giving you a confident paragraph, it opens scientific papers, checks a protein structure, generates molecules, docks them, scores the results, and then says which experiment should come next. That is the shift behind NVIDIA BioNeMo Agent Toolkit. The interesting part is not that an AI can talk about biology. The interesting part is that it can be connected to real scientific tools, and that creates a new question. When does a chatbot become something closer to a research assistant? For years, the popular image of AI drug discovery was simple. Train one powerful model, feed it a molecule, and let it predict whether that molecule might work. Maybe the model predicts toxicity. Maybe it predicts binding. Maybe it designs a new chemical structure. That is useful, but it is also a narrow slice of the real work. Drug discovery is not one question. It is a long chain of questions. What biological target matters for a disease? Is that target actually involved in the disease, or just correlated with it? What protein shape should we use? Which molecules might bind to it? Will those molecules dissolve, enter the right cells, avoid the wrong proteins, and survive in the body long enough to matter? And after all of that, can a lab actually make and test them? The old bottleneck is iteration. Biology has an enormous search space, and wet lab experiments are expensive, slow, and limited. A scientist does not want one pretty molecule. A scientist wants a loop that can narrow the search, explain why, run the next check, and keep track of what failed. This is where agentic AI changes the mental model. A normal AI model is like a specialist sitting in one room. You bring it one input, and it gives back one output. An AI agent is more like a coordinator. It can break a goal into steps, choose tools, call those tools, inspect the results, and decide what to try next. In ordinary software, that might mean reading files, writing code, or querying a database. In life sciences, the tools are different. The agent might call a protein folding model, a molecular docking tool, a genomics pipeline, a molecule generator, a literature search system, or a scoring model for drug-like properties. BioNeMo Agent Toolkit is NVIDIA's attempt to package those scientific capabilities so agents can use them more reliably. NVIDIA announced it on June 23, 2026, and positioned it as a domain-specific tool layer for life science agents. The company named organizations, including Lilly, OpenAI, Anthropic, Schrödinger, Snowflake, Databricks, Dassault Systems, and the Institute for Protein Design at UW-Medicine as adopters or integrators. That matters because the AI scientist idea often sounds like a demo. A model answers a biology question in a chat window, and everyone imagines an automated Nobel Prize. But infrastructure is less glamorous and more important. The hard part is not making the AI sound scientific. The hard part is giving it safe, documented, repeatable ways to do scientific work. Think of BioNeMo Skills as instruction cards for scientific tools. A skill tells the agent what the tool is for, what inputs it needs, what parameters matter, what files or artifacts it should expect, and what kinds of failures to watch for. This is very different from giving an agent a random endpoint and hoping it figures everything out. A beginner-friendly way to picture the workflow is a research loop. First, the agent gathers evidence from literature and datasets. Then it chooses a biological target, maybe a protein involved in a disease pathway. Next, it uses a structure model to estimate or inspect the shape of that protein. Then it generates or retrieves possible molecules. After that, it docks those molecules into the protein structure to estimate possible binding positions. Then it scores and filters the candidates. Finally, it recommends the next computational check or lab experiment. The point is not that every step is perfect. The point is that each step produces something inspectable. A paper summary can be checked. A protein structure has confidence signals and files. A docking run produces poses and scores. A generated molecule can be tested against chemical filters. The agent is useful when it keeps the loop moving while leaving evidence behind. If this kind of AI explanation is useful to you, consider subscribing. We focus on the mechanisms behind AI, crypto, and future technology without the hype fog. Now back to the machinery. One key piece in NVIDIA's approach is NIM, which packages models as callable microservices. In simple terms, instead of every lab manually installing complex research code, dependencies, model weights, and GPU settings, a model can be made available through a more standardized service. BioNeMo then adds the agent-facing layer on top. That layer is important because scientific tools are picky. A protein sequence might need a specific format. A molecule might be represented as a SMILES string. A structure file might come back as a CIF file. A docking model might require a prepared protein pocket and ligand input. If an agent gets any of that wrong, it may still produce confident text, but the science underneath can be invalid. This is one of the biggest misunderstandings. The frontier language model is not the whole AI scientist. The language model is closer to the planner and explainer. The scientific models and workflows are the instruments. A good agent system connects the planner to the instruments in a controlled way. An analogy is a hospital. A doctor does not diagnose only by talking. They order blood tests, imaging, biopsies, and specialist reviews. The doctor interprets evidence, but the evidence comes from tools and procedures. An AI scientist works in a similar pattern. It should not just improvise biological claims. It should ask for measurements, run analyses, compare outputs, and explain uncertainty. Now, let's make this concrete. Suppose a research team is interested in a protein target. A basic chatbot might summarize the target and suggest some well-known molecule classes. A single prediction model might evaluate one candidate at a time. An agentic workflow can do more. It can retrieve known information about the target, choose a folding or structure prediction tool if needed, generate a set of candidate molecules, dock them, rank them, flag weird results, and propose which candidates deserve deeper simulation or wet lab testing. This does not mean the AI discovered a drug. It means the AI helped organize a search. The difference is crucial. A drug is not a molecule that looks good in a computer. A drug has to work in a living system, at a useful dose, with acceptable safety, manufacturability, and clinical evidence. Computational discovery is a filter, not a finish line. That brings us to the failure modes. The first failure is hallucinated biology. A language model can connect concepts that sound plausible, but are not supported by evidence. In biology, plausible is not enough. If the agent invents a pathway, misreads a gene function, or treats a weak association as causal, the rest of the loop can become beautifully automated nonsense. The second failure is the bad tool call. The agent may pick the wrong model, pass inputs in the wrong format, use the wrong biological assumptions, or misinterpret the output. A docking score, for example, is not a guarantee that a molecule will work in a cell. A predicted structure may be uncertain in the exact region that matters most. If the agent treats every number as truth, it can amplify errors quickly. The third failure is false positives. Drug discovery already has many molecules that look promising early and fail later. AI can make this worse if it generates huge numbers of candidates that satisfy easy computational filters while missing harder real-world constraints. More candidates are not automatically better. Better triage is the goal. The fourth failure is automation bias. When a system produces a clean report, humans may trust it too much. This is especially dangerous when the report includes technical language, charts, and rankings. The more professional the output looks, the more important it is to ask what evidence supports it. That is why humans and labs remain in the loop. Scientists still define the question, judge the assumptions, design the experiments, and decide what evidence is strong enough to act on. Labs still test whether the molecule behaves in the real world. Regulators still require evidence for safety and effectiveness. An AI scientist workflow can compress parts of the search, but it does not repeal biology. The deeper mental model is that these agents are becoming research operating systems. An operating system does not replace every app. It coordinates files, permissions, memory, devices, and processes. In the same way, a life science agent can coordinate data, models, simulations, lab systems, and reports. This is why tool packaging matters. If every scientific model is a separate island, only experts can use it well. If models become documented, callable skills, then agents can chain them together. That makes workflows easier to reproduce, easier to inspect, and easier to improve. The big shift is from one model answering one question to a loop that asks, tests, learns, and asks again. In drug discovery, that loop might move from literature to target, from target to structure, from structure to molecule, from molecule to score, and from score to experiment. The agent is not magic. It is the coordinator of that loop. BioNeMo Agent Toolkit is important because it makes this shift visible. It shows how AI drug discovery may become less about isolated model demos and more about connected workflows. The future value will not come from chatty systems that sound like scientists. It will come from systems that can use the right tools, produce checkable outputs, expose uncertainty, and help human researchers make better next step decisions. So when you hear the phrase AI scientist, do not imagine a machine secretly curing diseases alone. Imagine a research assistant with access to a growing toolbox. Sometimes it will speed up a search, sometimes it will make mistakes. The difference between hype and progress will be whether the loop is grounded in evidence. And that is the real story. AI in drug discovery is moving from prediction to orchestration. The question is no longer only, can a model predict this molecule? The better question is, can an agent manage the scientific loop well enough that humans can discover faster with clearer evidence and fewer wasted paths? If you have questions about BioNeMo, AI scientists, or how agentic workflows work in biology, leave them in the comments. And if there is another AI or biotech topic you want explained next, suggest it below.